everyone. Welcome to the 30th edition of Soup and Science. My name is Preeti and I'm one of the organizers for this event. Before we begin, we acknowledge that McGill University is on land which has long served as a site of meeting and exchange amongst Indigenous peoples, including the Anishinaabe and the Haudenosaunee nations. We acknowledge and thank the diverse Indigenous people whose footsteps have marked this land on which peoples of the world now gather. I'm going to turn the microphone over to our fabulous host, Professor John Sticks, who is the Associate Dean of Research. John will introduce the speakers for today.
Thank you very much, Preeti. I appreciate the kind words. And we're very excited today to have four wonderful speakers with us. Karine Auclair in chemistry, Debra Titone in psychology, Anush Tserunyan in math and statistics, and Mathilde Rioux in psychology. So when the presentations are finished, you're going to receive a message from the host to join a breakout room. And in this breakout room, you'll be in smaller groups and you'll have the opportunity to ask questions to each presenter. And each presenter will rotate through the rooms every 10 minutes until they have visited all three rooms. And then at the end, we'll return to the main session for some final remarks. So I'd like to introduce our first speaker, Professor Karine Auclair in chemistry. She's our CRC's Canada Research Chair, Tier 1 in antimicrobials and green enzymes. And I think maybe the best way to describe her fascinating research is that she uses chemical tools to understand biological systems. So welcome, Karine. We're very pleased to have you with us.
Thanks very much, John, for the introduction. I had some problems. It cut. And I hope that you guys will be able to hear me well. Everything is frozen again, sorry. Pretty.
Is that better?
Give me a second, it's frozen.
Yep. Okay.
I think it's good now. Okay, so now I'm gonna start. Good morning, everyone. So the research in the Auclair lab focuses on harnessing enzyme to make a better world. We have interest to develop new processes that are more environmental friendly, where we change harsh chemicals for enzymes that work under mild condition. We also have interest in blocking bacterial enzyme to make new treatments for infections. Sorry, the slide doesn't change. All right, there we go. So enzymes are very, very large molecules that are produced by all microorganisms. They are the chemists of our cells. They're renewable, non-toxic, they work under mild condition, and they're selective, and they catalyze reactions. But they're also involved in diseases and infections. So one of our goals is to block bacterial enzyme to develop antibiotic resistance inhibitors, as well as new antibiotics, but also molecules that don't kill bacteria, but rather resensitize them to the host immune system. We call them bacteriomodulators. I won't tell you more about these because I don't have time. Today, I want to focus on our processes, new environmental friendly processes. Here we start with waste from agricultural industry or from the forest industry, as well as waste from the fishing industry like crustacean shells, as well also as the most talked about waste nowadays, and sorry, my slide is not changing. Okay, so the most talked about waste today, which is plastics. All of these wastes are polymer molecules that are very unreactive. Oops. I'm having a hard time changing the slides. I don't understand. I'm really sorry. So these wastes are all very large molecules that are polymeric. So they're very long molecules that are very unreactive and they're insoluble. So they're difficult to break down. And chemists have developed methods to break them down. However, these methods require lots of solvents, high heat, toxic reagents. They may also need like extreme high heat, like in pyrolysis, with all the problems that come with it. And a few people have developed enzymatic processes, but those require large volumes of water and also require that the polymers are pre-treated with harsh reagents. So we want to do things differently. Typically, enzymes are used in very dilute aqueous conditions by us. However, in nature, enzymes are in a very crowded environment with lots of molecules around and not that many water molecules available. Another example is fungi that grow on tree. They actually spit out enzymes which actually work on the surface of the tree, so they're not in water at all. So we propose to work with moist solid enzymatic reaction mixture. But how do you work with these solids? So the way that we shake these mixtures, it should actually use milling, ball milling, but there are many other ways. So here we start with the solid enzyme, the solid waste. We introduce that into a milling jar with steel balls, and then we close the milling jar, and then we hook that into a mill, which is going to shake it. So we don't use solvents, we don't have solubility issues, we don't need to heat, and there's no toxic reagent. Now, the big secret of our method is raging. Raging is the fact where we shake a little bit, and then we let it sit. We call this aging. We shake a little bit, we let it sit, and we repeat this process multiple times. With that, we can actually transform forest waste as well as agricultural waste into glucose, which can be used to make bioethanol or other molecule. We can turn crustacean shells into acetylglucosamine, which is an important precursor to nitrogen-containing molecules. And we can turn plastic waste into valuable chemicals or enable the true recycling of plastics. So I'm leaving you on this slide that has a picture of our group and our lab, as well as our website and our Twitter account. Thank you. Again, I apologize for the delays with my slide.
Thank you so much, Karine. That was really interesting. I'd like to introduce our second speaker, Debra Titone. And Debra is Canada Research Chair, Tier 1 in Language and Multiculturalism. And she's interested in, among other things, language processing and bilingualism. So welcome, Debra, and we look forward to your talk.
Thank you. Can you all hear me? And can you see my cursor moving on the screen? Yeah? Okay, fantastic. Yes. So thank you for inviting me to this. I'm very excited to just give you a quick overview of my research. As I'm talking, I'm hoping that maybe all of you who are out there can maybe type in the chat and tell me all the languages that you know, if you feel comfortable doing that. And then I'll start speaking, okay? So in the Language and Multilingualism lab, I'm trained as a cognitive psychologist, cognitive neuroscientist, and a psycholinguist. So we're interested in how people who know multiple languages, which I can see as the chat unfolds here, many of you do, how do you manage your competition that might occur between the languages that you know in everyday conversation or reading or things of this sort? And of course, we also know that not all bilingual experience is the same, right? So for all of you, like look at all in the chat, all the different configurations of languages that are appearing here. Some of you might have learned your languages simultaneously in the home. You might have learned your languages sequentially. You might use your languages in different ways throughout your daily experience. All of these are potentially relevant for how use of your languages can shape the neural structures supporting cognition and how you process language in real time. So the core research questions of my lab revolve around how do people manage competition between their languages? What are the relevant individual differences in bilingual experience that might matter? And what are the broader neurocognitive consequences of managing two language systems? Now, this is going to be really tricky, right? Because how people use their languages is very social and it's very messy. So one of the things we've been trying to do is find ways to quantify these complex experiences that people have. But before I give you a couple of examples of that, I just want to tell you about the kinds of methods that we use in my lab. We use a broad array of methods ranging from behavioral, plain old button press types of experiments, cognitive psychology, We use eye tracking to a great extent to examine how people read naturally. So we could present sentences on the screen and track where the eyes fall on specific words. Less frequently, but sometimes we use neurocognitive measures like event-related potentials, where we can measure the neurophysiology of the brain as it's engaging in language processing, and also neuroimaging tools. So we use a broad array of tools and techniques to get at the kinds of questions that are important for how bilinguals and multilinguals manage the languages that they know. So just to give you kind of a flavor of what we've been doing, this gentleman right here is Dr. Jason Gulliver. He's a senior postdoc in my lab. And he and I, across a number of papers, have been developing this a notion of language entropy. So what we're trying to sort of capture quantitatively is how people might distribute the use of the languages that they know across social experiences in their daily lives. So for example, imagine this guy here. He knows, both of these people know English and French, but this person tends to use English most of the time, whereas this person tends to use both English and French. Well, it turns out to quantify that, we can use this mathematical concept of entropy, which has to do with certainty of a particular construct versus uncertainty. So if somebody uses a single language in most contexts, there's going to be a high degree of certainty, therefore entropy is going to be low. And if somebody can use multiple languages in any context, there's a certain amount of uncertainty about what language they're going to use, so entropy would be high. So we've been trying to validate this measure across different tasks and different levels of analysis to see if it has psychological reality. So that's just one example of what we've been doing. Another example of what we've been doing, again, is trying to quantify what do bilinguals and multilinguals talk about in their different languages, right? So often what we study in cognitive psychology and cognitive neuroscience is the how of how people process language. But what we're trying to get at as well, because we think it's important, is what they talk about. So in a recent paper that's now in press, we've been using the tools of network science, which is another kind of quantification to get at this idea of what people say, what people talk about in their different languages. So network science, basically you develop these mathematical formalisms where you've got nodes and edges. And in this particular paper, what we did, I just want to also say this work has been led by a doctoral student in my lab, Mathilde. Jason Gulliver was involved with it, and an undergrad who did research stints in my lab is also on this paper. And all three of these students were absolutely instrumental in getting this work going. So with network science, we can conceive of nodes and connections. And in this particular study, the nodes represent the topics of conversation that people can have. And the edges, which are the lines connecting the nodes, could either represent the social context of where these topics are communicated about, or the languages that people are using to communicate about these topics. So for example, if we look at the results in terms of topic networks by language, remember again, the little dots represent the kinds of topics that people are talking about, whereas the lines represent the what's talked about in the same language. So this has to do, so this formalism here gives us a picture of what are people talking about in their dominant language, what are people talking about in their non-dominant language, and the purple reflects the fact that when in the dominant language, people are talking about a broader range of topics and social experiences, whereas in the non-dominant language, people are talking about a smaller array of topics and social experiences. Now we could flip this formalism, and we could now plot it where the dots again are, the nodes again are the content of the conversations or the communication, and the lines represent whether people are communicating in one language or multiple languages. And then we can do this plot across social contexts or across social contexts. And what we can see here is that this, as we move across these social contexts, the number of languages used goes from small to large, right? So in the work context, it says yellow. So yellow means that there's fewer languages used in the work context. And then as we go from work to school, you start getting a little bit more green in there, right? So the idea being that people are using their languages systematically in different ways across different social contexts. So what we hope to do with these types of formalisms, both the language entropy and these sorts of quantifications of network models and things like this, is to now take this as individual difference characteristics that we can then link to other cognitive measures. So for example, how do people comprehend spoken language in their first and second language? Or how do people deal with syntactic ambiguity or other types of language processing characteristics? And finally, also, we're interested in this kind of question that's out there a lot in the news now about whether bilingualism or multilingualism makes you smarter, right? So there's a lot out there now where people are trying to make the case that the more bilingual you are, the better you are at general cognitive capacities like executive control. Well, how we approach that question in our lab is from the vantage point that not all bilinguals are the same, not all multilinguals are doing the same thing. So by having these sorts of sophisticated formalisms that can characterize bilingual experience, we could be in a better position to make linkages between language processing and general cognitive function. So that's all I have for you today. I want to thank the funding sources. This is a picture of my lab. This is like one of the first Zoom meetings when the pandemic started. And we're growing. This was just earlier this week. So thank you very much.
Thank you very much, Debra, for an amazing talk. And I was wondering, maybe you have an extra place for me in your lab. That would be great.
Oh, come on in. We'll put you in.
Thank you.
You'll be in the Zoom array.
Thank you very much.
Okay.
So our next speaker is Anush Tserunyan in the Department of Math and Statistics. She's an assistant professor there. She has a number of research interests, including descriptive set theory, also combinatorics, which address ways of counting, and finally ergodic theory. And I think she's going to talk to us about ergodic theory. So better she explain it than I. So welcome, Anush. Thank you very much for being here today.
Thanks, John, for the introduction, and thanks, Preeti and everyone else involved for the invitation. So yes, I'll talk about ergodic theory today. So ergodic theory is a study of so-called dynamical systems. So what is a dynamical system? It's some sort, I put some pictures. So a dynamical system is a space on which there is some action. So there is a transformation, which I denote by T for transformation. So this transformation T moves points of X to other points of X. And in fact, it's perfect. In Debra's talk, Shannon entropy is defined for dynamical systems exactly. So this works great. And so I put some, so for instance, I can explain the second picture here, which is a picture of a coffee. So stirring coffee is a dynamical system. The space is coffee itself. And when you stir, you move points of coffee to other points of coffee. So particles of coffee get moved around. A simplest, formal mathematical example is rotation of a circle. So let X be the circle here. And T rotates the points of circle by an irrational angle, more precisely by an angle alpha times pi, where alpha is irrational. I choose it to be irrational so that when I rotate, I never come back to the same point, so that the dynamical system is interesting. So you can see if I take point X here, rotate by this angle alpha times pi, I jump to T of X, that's where T takes X. And then afterwards, if you apply T again, you go to another point, which is T square of X over here. And then when you apply it again, you go to T cubed of X and so on. And so it keeps rotating if you keep iteratively applying T. And so the main concept about one important concept of dynamical system is that of ergodicity. So roughly speaking, a dynamical system is called ergodic if the transformation T stirs it up really well. What do I mean by this? I mean this, again, intuitively. If you first put milk in coffee, before stirring, you can see the differences. There's part of coffee that's with milk, another part that's without milk. But once you stir, after some finite amount of time, very short, your coffee is homogeneous. You have all parts of coffee have milk in it. More more formally speaking, mathematically, how do we define this like stars up very well? The way we define it is as follows. You say a dynamical system is ergodic if whatever region you pick, so here, look, here is this my irrational rotation. I pick some region in it, say this blue region. So whatever region you pick and fix, then every randomly chosen point after iterative applications of the transformation will land in this region, in this blue region you chose. So here you can see you start with X, and it, you know, t to the 7 of X already visits this region. So basically every randomly chosen point has to visit the fixed region that you chose in the beginning. And as opposed to irrational rotation, so irrational rotation is ergodic. One can prove this, but here is sort of intuitively why it happens. But rational rotation is not ergodic because irrational rotation, every trajectory of a point is finite, and there are lots of big regions that you will miss and never visit. Okay, so ergodicity is like well mixed, well stirred. Non-ergodicity is the opposite of that. Now, how is this ergodicity used? So let's say we want to compute an average value of some function that is defined on our space. And average value, well, you're all scientists, you know, it's the integral of that function. For example, let's say we take the circle example from before, think of it as the equator of Earth, and let f be the temperature function. And it's natural to want to know what is the average temperature of the equator. So note that, okay, before I do the theorem, note that X is very infinite. Our space of points is very infinite. In fact, there's a continuum of them. So it's really hard, like how are we going to measure infinitely many points, temperature, and find the average? Well, we don't because of this theorem. So it's called pointwise ergodic theorem. It's due to Birkhoff, and it bridges it makes a bridge from discrete measurements to continuous. So that's why I entitled my talk from discrete to continuous. So what does it say? It says if your dynamical system is ergodic, so well mixed, then average of any function, this integral of any function, can be well approximated by finitely many measurements, and you take average of these finitely many measurements. What are the finitely measurements? You begin with any randomly chosen point X, you measure the function at X, then at T of X, and so on at T to the n-1 of X, and divide by the number of measurements. And this number magically will give you a very good approximation to the integral. And as n increases, the approximation gets better. So I guess this is the main chunk of the talk, the advertisement of this theorem, which dates back to Birkhoff, actually, from 1931. So it's a very old theorem which started the subject. But today, okay, so what does the subject do today? For instance, why consider only one transformation? We may consider many, and in fact, systems are such that many transformations are acting. For example, on the torus, also known as the donut, you can easily create two transformations, which are two rotations around each axis, okay? So this would be two. How do we have so the question is, do we have an ergodic theorem, pointwise ergodic theorem for this kind of systems? Note that when we had a single transformation, if you look at the trajectory of one point, then you can model it by a discrete line, because you have X, then you have T of X, T squared of X, T third of X, so it's like integers. Whereas
you have about 30 seconds left. Sorry to interrupt. 30 seconds left.
Okay, thanks. Whereas when you have two transformations, it's more like a four-branching tree, whose picture I put here. So in a very recent work with my PhD student, Jana Zombek, we are able to prove a pointwise ergodic theorem for any number, multiple transformations, where the pointwise averages are taken over finite subtrees of this pretty picture that I put. That's it. Thanks for attention.
Thank you very much, Anush. I think I have about 10 questions that I'd like to ask you, but I don't think I have time for that. So our last speaker, last but not least, thank you. Last but not least, our last speaker is Mathilde Rioux. She's an undergraduate student in the Department of Psychology. and she is working in, if I'm not mistaken, in Signe Sheldon's group in what is called the Memory Lab. So over to you, Mathilde. Welcome to Soup and Science.
Thank you very much. So today I will talk about one of the projects that I did at the Sheldon Lab last year. So it's my first honors thesis, which I had the pleasure to do with Laurie Gergian, who is a graduate student of Dr. Sheldon. And what we wanted to look at was how rehearsal could change the neural representation of our memories. And more specifically, we focus on autobiographical memories, which are internal representations of past personal events. So every time these memories are remembered, they are reconstructed in the brain, more specifically in the hippocampus, which is a brain region known to be very important for remembering. And theories of functional specialization have indeed suggested that possibly the anterior and the posterior part of the hippocampus have different functions for remembering. So the anterior hippocampus might be more important for retrieving coarse-grained details associated with a specific memory, whereas the posterior hippocampus might be more important for retrieving the fine-grained details associated with such memories. And one of the factors that can affect neural activity in the hippocampus is rehearsal. So this is basically when you think about the same memory, so when you retrieve the same memory multiple times throughout a certain period of time, this would be an example of rehearsal. And past studies have shown that usually through rehearsal, you see a decrease in neural activity. So here our objective was to investigate how rehearsal would alter the support for remembering autobiographical memories. And we expected to see a decrease mainly in the interior hippocampus because of its role for the initial construction of our memories. Oops. Oh God, sorry. Okay, so how did we go about testing this? We recruited 24 participants who were asked to complete an autobiographical memory questionnaire in which they provided us with 24 past personal events. From those, we selected 12 memories based on how similar they were to each other. And then during the scanning sessions, when participants came in, we asked them to lay down in an fMRI scanner so we could record their brain activity. And when they were laying down, they had a screen on top of them in which a small caption would appear, and each caption would refer to one of their 12 memories. So the caption for each memory was shown 4 times, and here we were interested to see how activity from time 1 to time 4 would change. So for example, here I call this guy Mark, who's laying down in the fMRI scanner, and he's shown the caption he previously sent to us about the first time he saw his dog at his 10th birthday. So he's told to think about that event 4 different times throughout the course of an hour. And here we look at how activity in the hippocampus specifically changed as the event was repeatedly retrieved. So what we found was a significant decrease in both the right and the left anterior hippocampus, but not a significant decrease in the bilateral posterior hippocampi, thus confirming our hypothesis. So the aim of our investigation was really to characterize changes in neural activity that support the retrieval of autobiographical memories. And we were able to show evidence of a repetition-related decrease in activity only in the interior hippocampus, which suggests that repeated retrievals do alter the constructive demands, and mainly it causes a lowered constructive demands of autobiographical memories. So please come by if you have any questions about either my research project or about doing research as an undergraduate student at McGill. Thank you.